Hello, everyone. First of all, thank you for this opportunity to be here. It's a big, big honor for me. And second, it's really true about the fish chamber turn into the growth chamber. This is happy me that I'm being able to grow plants in the fish facility. And I will start with this question. Do you think that we can currently predict the outcome of adaptive evolution? There was a great symposium about it yesterday. I think the answer was it's complicated and we don't even know what the predictability is, or we don't have it clearly defined. And today I'm not going to give you any definite answer, but I would like to offer you my way on how I like to look into this question. So I like to look into the predictability of adaptation by studying its repeatability. And by repeated adaptation, I refer to the phenomenon in which multiple different lineages independently increase their fitness under similar conditions. Textbook example of this is the three-spined sticklebacks, which is, by the way, the fish which I outcompeted in Katie Peichel's group. They are originally marine, but they repeatedly colonize fresh waters across the globe and independently adapted to these instances of fresh waters. And you can study the repeated adaptation at very many levels, thanks to Mehdi, who made the illustration, starting at the mutation up to the phenotype. And I am the most interested into the repeatability at the genetic level. So asking how frequently and why species reuse their genes in these independent instances of adaptation. So just again, one example from sticklebacks. If we look into the signatures of selection associated with freshwater colonization in America, we would find this IDA gene, and the same holds true for the colonizations across Europe and all the different places. So this would be a gene reuse by the IDA gene. Simple. And I see two main approaches how you can think about the repeatability of genetic basis of repeated adaptation. First, which I call reverse genomic approach here, would require you to go out, sample as many independent lineages repeatedly colonizing similar environments as possible. And then by analyzing their genomes, you can ask which genes or phenotypes are repeatedly involved. With the forward genetic ones, on the other side, you start with a phenotype, which should be ideally some phenotype which is repeatedly emerging and which you find important for the adaptation, and then try to see how frequently do species reuse genes to make this phenotype and what makes these genes to be repeatedly reused. And both of these approaches also allow you to ask slightly different questions regarding the repeatability of adaptation. So I think that the reverse genomics approach is great for comparing the repeatability among different lineages or species, asking why some species adapt more similarly than others. Whereas the forward genetic one allows you to compare genes, so asking what makes this gene so special that I see it being used by evolution over and over. And in this talk, I want to show you how I combine both of these approaches to learn more about the adaptation. So I will start with the reverse genomic ones, where I decided during my PhD to test this very, very intuitive hypothesis that the more are certain species or lineages related, the more gene reuse they would have during their adaptation. And the goal wasn't to really just test if this relationship exists, but the main purpose was to ask what might be the underlying mechanisms. And I didn't use fish, as you already know, but I used plants, specifically the model system of alpine adaptation in the genus Arabidopsis, but not the big plant model Arabidopsis thaliana, but other species. You will see this soon. Why did we choose alpine environment for our study? So we had two reasons. First, it represents a set of well-defined challenges for plant life, including cold or freezing temperatures, high UV radiation, fluctuation, precipitation, different herbivores, and so on. With the freezing temperatures comes also the fact that there is a very short summer season, which is a stressor by itself. This is very intuitive, but coming from relatively lowland Czech Republic, we were many times badly surprised about how short the summer season actually is. And the second reason why we chose alpine environment is that mountain ranges are distributed as kind of islands in otherwise dominantly lowland landscape. And this may trigger the repeatability of adaptation. And we can see it both if we just look into the landscape, but also at the distribution map of the alpine ecotypes. So here are some of the examples of how my model species, which the main one is Arabidopsis arenosa, looks like. It can be either tall, wide, biennial, foothill, which you see at the bottom row, and occasionally it forms these alpine ecotypes, which are short, pink, perennial, and there is many and many other traits in which they differ. And actually the alpine populations or ecotypes are so similar to one another that it fooled 19th century taxonomists into naming them as a separate species, Arabidopsis neglecta. But now, thanks to our genetic analysis, we know that the alpine ecotype is always more related to its geographically proximal foothill plant than to the other alpine ecotypes. So this is a nice signature of repeatability. And here I show 3 different examples of such foothill alpine lineages, but we actually discovered 5 of them in Arabidopsis arenosa, and also 3 more in its closely related species, Arabidopsis halleri. And I think 7 lineages is already quite a nice data set to test this hypothesis about the role of relatedness in the repeatability of adaptation. So what do we need if we want to test this hypothesis? So first of all, we need that our sets of repeatedly emerging lineages have variable divergence times among them. And you can see on the phylogenetic tree that this is the case in the Arabidopsis system. They range from the yellow ones, which are relatively recent, up to the different species level. That would be the orange one. I can't see it here on the screen, so it's a bit confusing. But and second thing, when you have the different divergences, it's good to have whole genome resequencing of the populations growing in the stressful environment. So in my case, the alpine ones, and also the foothill ones, just as a contrast. And then you can apply some kind of selection scans or environmental association to find your candidate genes and finally overlap them to see what's the degree of gene reuse among these instances of repeated adaptation. And maybe just a side note to the divergence, this doesn't refer to the length since the onset of selection, so the length for which my populations would sit in the Alps. These are roughly the same, they are postglacial, but this refers to the neutral divergence of the lineages before they even colonize alpine environment. And well, I show this very simple Venn diagram, but in reality, if you have 7 lineages, you get such a crazy sets of combinations and overlaps. So that would be a bit overwhelming to interpret. So I decided to work in a pairwise manner, comparing the 21 possible pairs which you can make among the 7 lineages. So now the results. Are you curious? Do we see the relationship? Thank you. You're a great audience. So in this graph, on the x-axis, we see the different divergence time categories. The yellow would be the shortest one, so the ones on the very left of the phylogenetic tree, and the orange would be the between species comparisons. And you can see this very obvious and also highly significant decrease of gene reuse with increasing divergence time. It's nice. It's also maybe not that surprising, but the interesting question is, why do we see this relationship? And because it's well established that the availability of genetic variation promotes adaptation, it was very intuitive to first ask whether the availability of shared variation between the lineages would promote the repeatability of adaptation in this system. So now the question is, how can two lineages share their adaptive variation or have the same adaptive variation? So there are basically three different mechanisms. First, it could be present in the common ancestor of both and then be inherited as ancestral standing variation, or it can originate in one and then be introgressed into the other, or it could mutate independently in both of the lineages. But because the independent de novo mutations are less likely than the presence and single origin of the allele, also the gene reuse, it's less likely for that scenario. However, as we increase the divergence time between the lineages, maybe hybridization barrier accumulate, so they would lose their capacity to share alleles via gene flow. And later on, also all the ancestral variation would be lost. So they can only rely to these less likely de novo originating alleles, and that would drive the decrease of the gene reuse among them. So this was my expectation. Now how to test for it. So for each divergence category, I took all of the reused genes which I had there, and I modeled what's the most likely scenarios for the origin of their adaptive variation. And this is summarized in this graph. In dark blue, you can see the proportion of the genes which were reused due to the presence of introgressed allele, so the gene flow scenario. The light blue would be reused due to the presence of ancestral standing variation, and the rest, summing up to 100%, would be the de novo origin. And you can see the clear decrease of the two allele sharing scenarios with increasing divergence time. So then I was able to statistically show that this decrease of allele sharing is actually explaining most of the variability between the divergence time and gene reuse. So this is likely the underlying mechanism for these recent timescales. So to just wrap this up a bit visually, as the alpine lineages of Arabidopsis increase in their time, the degree of allele sharing is decreasing, and this is in consequence driving the decrease of gene reuse. And with this story, I basically finished my PhD under Philip Collard in Prague. Here are just a few illustrations how it went. It was a lot of fun. And I decided to move to Switzerland, to Katie Peichel's group, because I think we shared a lot of conceptual thinking about the repeatability of adaptation. And the first obvious thing to do there was to ask, is this really a general trend, or is it just some weird thing which we see in these alpine Arabidopsis lineages? So for that, we decided with Katie we would review the available literature.
Timer's not working.
But I have 2 slots, so no stress. Thank you. So we would review the available literature and summarize, searching for the studies which are studying repeated adaptation across a divergence scale, and we would summarize whether they show the same pattern as my alpine Arabidopsis. These would be summarized in this green upper triangle. If they don't show any relationship between divergence time and gene reuse, that would be the gray, or even the opposite relationship, that's the red. It's a complicated scheme, but I think we can read 2 things out of it. So first of all, if we look at the more recent time scales, so roughly the divergence time between different lineages, populations, and species, we see a strong support for the negative divergence dependency. And interestingly, the borderline between this is roughly at the time scale where we think, which is like different closely related species, where we don't expect there would be any more allele sharing above this. And if we look for the older divergence scales, we don't actually see support for any of these scenarios. And this was a bit surprising to us because we identified other mechanisms in the literature which might drive the divergence dependency of gene reuse also across very broad timescales, like the differentiation of genome architecture, of gene functions, different phenotypes being used for the adaptation, etc. But the trend is just not yet present in the literature. We can discuss it together after the talk. We also wrote a review about it with Katie, which just recently came out in Tree. And if you are interested in the mechanisms or in our discussion why we do and don't see this relationship, you are welcome to read it. Here I give you a few moments to digest this complicated scheme. And I have a drink if I find my bottle. Which I don't, never mind. It's fine, I don't need to drink. And I will jump to the second approach, which is the forward genetic approach. And there, it allows us to ask what determines the reuse of different genes. And there could be very many factors, but here I want to focus on two, and that is the increase of probability of gene reuse due to the constraints, due to the pleiotropy, and also due to the availability of shared variation. And as the candidate phenotype for this approach, I selected the pink petal pigmentation, again in my model species Arabidopsis arenosa. So in this distribution map, you can see how the different petal colors are distributed throughout the Central Europe. And we can see two things there. First, the Arabidopsis arenosa is predominantly white, but there are patchy distributions of the pink-petaled populations, which mostly coincide with the alpine environment. And second, you can see that despite this is in the lowlands, it's dominantly white, you can see tiny bits of the pink already in the populations proximal to the alpine ones. So there is probably standing variation for this trait already available in the foothills. We see similar things on the phylogenetic tree. From there we can read that the white color is ancestral, the pink one is emerging repeatedly, it's associated with the alpine environment, and you can see that there is a bit of pinkness in all the lineages sister to the alpine lineages. Alpine ones are those circled by black. I hope you got it from the legend. And what is also nice about this distribution map, that anytime I plot the bigger pie chart there, it means that we have a population-level whole genome resequencing data for that population. So that allowed me to do two things. First is the GWAS for the genetic basis of the pink pigmentation. And second is the genome scan, where I compared all of the fully pink populations to all of the fully white populations. And these two analyses agreed on one peak. And when I zoomed into it, there were two genes. And here comes a really nice surprise of this project. When I looked into what are the genes, the first one was called the production of anthocyanin pigment 2. And anthocyanins are the compounds which are making the petals pink. So this was really nice. That's the time point when I needed to turn the fish room into the growth chamber, because I wanted to see if I would be able to map this genetic basis also in the lab. I did a series of experiments, and here I show you just one of them, in which I decided to do a large-scale screening of the seeds from the fully pink alpine population. And I was trying to identify a rare heterozygous plants which would bear both the pink and the white alleles. Then I crossed them together, and I was very pleasantly surprised that the top, I was able to make a white flowering alpine plant. You can recognize it's alpine because it's super short. It doesn't have any inflorescence stem. And this white form was segregating in the Mendelian way together with the light pink and normal pink. I'm now planning a series of follow-up experiments on this, trying to understand what could be the functional meaning of the darker pigmentation in the alpine zone, and also what could be the potential fitness advantages of this phenotype. And if you want to discuss it, I would be super happy, because I have a lot of ideas, a lot of negative data, and it would be good to know what do you think. But this is a bit of detour. Now let's come back to the main line of this presentation. So, do we see any gene reuse for the evolution of the darker pigmentation of petals? Thankfully, there is a huge body of literature mapping the genetic basis of petal color pigmentation due to the anthocyanins. I knew about these studies, I knew that there are various loci identified, and I knew they sound different than my PAP2 gene. But what was a second super nice surprise of this project is that when you do a phylogenetic tree of these different loci, you discover they are all orthologs of the same gene. So basically here, across the evolution of angiosperms, we see a very high degree of gene reuse for the evolution of petal pigmentation. And now the question is, what makes these PAP-like genes to be repeatedly used across such a broad timescales? And thankfully, there is also a lot of knowledge about that. And I think most of the studies agree on two things. First of all, they highlight that this is a transcription factor, so a regulatory gene, which might, if you modify it, break less functions than if you would be modifying the core biosynthetic pathway gene. And second, this transcription factor is affecting the downstream genes in the anthocyanin pathway. So again, by modifying it, you won't break any upstream functions like the productions of flavonols on the left. So good, almost there. Both of these facts suggest that there is a low constraint due to the pleiotropy, and that could be why this gene was so frequently reused. Also within my Arabidopsis species, I think the repeatability is mostly due to the standing variation in the system. They would just repeatedly sweep up the variant which is already present everywhere in the foothills. So to wrap this up, first of all, we saw, and sorry, the pink pigmentation in this system is repeated, is associated with alpine adaptation, and its genetic basis is highly repeatable as compared to other systems across angiosperms. And this is probably due to the low pleiotropic constraints and high availability of standing variation at the within species level. And again, as previously, it's good to ask to which extent these findings about the low pleiotropic constraints would be generalizable over multiple model systems. So for that, we currently have a group project in the Katie's group. I see some of the participants over there, where we review different phenotypic changes, trying to see what's the level of gene reuse, making these changes, and try to understand what makes the reused gene to be special, what makes them to be repeatedly used by adaptation. So to conclude, throughout my very short research career, we were able to find some trends in the repeatability of adaptation. At the between species comparisons, we were able to see that the more closely related lineages would adapt more repeatedly. And at the genetic level, we would see that the pleiotropic constraints might increase the probability that certain gene will be reused. But despite seeing these clear trends, I'm not really sure that this means that adaptation is predictable. And I leave this open up to you, up to your own consideration. And with this, I want to thank the very many great people who helped me with all of these projects. I especially thank Katie Peichel and Philip Collard, who are both great, great, great advisors, and I'm very honored I could work with them and can work with them. And finally, one little personal side note. I also had these two little helpers, which are my sons, throughout most of my PhD and 2 postdocs. And I should finish, right? It's doable with kids.
So we have time for a couple of quick questions, if you could come to the microphones. Anyone?
I was just curious, that was a really nice result when you showed the pigmentation gene, but then you didn't say what the other gene was. Did you look into what that was, and does that show any similar patterns, I guess?
It's a calcium transporter, which doesn't sound very functionally relevant. But what is interesting is that is strictly, specifically expressed in petals. And now we know the anthocyanins are also needed in the leaves. So, and we know that the alpine plants are not getting more red in their leaves. So I think actually that could be some kind of contribution of both of the loci, kind of turning the tissue specificity of the PAP2 into petals. But that's very unclear, unstudied thing.
Sounds like an interesting idea. Thank you.
Thank you for asking that.
Thanks for your talk. Yeah, I was very small. Okay. So yeah, you were presenting the results of your literature review, and you saw something that you weren't expecting for lineages that diverged since a long time ago. And do you have any hypothesis of why that is? Yeah.
I think we don't study long enough time scales. We very recently cover within one study a system which would both have like between species divergence up to, for example, different kingdoms.
Okay.
And in the review, we are suggesting which selection triggers might be generalizable over these long time scales, and maybe if we study them, we might learn something more.
Thanks.
Thanks for the question.
Thank you, Magda. So I will say, not just Magda, but all 3 of our winners today all 3 of the JMS Prize winners are mothers. So Emily Roycroft is our next speaker. She is the runner-up for 2024 for the JMS Prize. She has done her work in, she is currently working as a postdoc in Craig Moritz's lab, but has already achieved a tenure-track position in Australia, which is the best prize of all. She uses genomics of both living organisms and museum specimens to study big questions in micro and macroevolution and using Australian rodents as a system. She also, it turns out, can raise the dead. She discovered that species thought to be extinct was actually hiding out under a different name and is still around. Are you able to get your slides working?
I don't know where the file is.
Oh, no.
Okay, thank you so much. I'm sorry that now we're a little bit behind, so I'll try not to take too long. Hi everyone, my name's Emily Roycroft. I work at the Australian National University in Canberra, and I'm going to talk to you today about some of our work looking at genetic purging in remnant island populations of threatened Australian mammals. I want to start by acknowledging that I live and work on Ngunnawal and Ngambri country, and that the work that I'm going to talk about today is based on specimens and species that occur across traditional lands all over Australia. And I want to acknowledge the traditional owners, the First Peoples of Australia, as the owners and custodians of those lands for time immemorial, and thank them for their ongoing custodianship and stewardship of country. Australia has the highest rate of historical extinction in the world, with 40 mammal species extinct since European colonisation of Australia in 1788. And that's for a number of different reasons, but some of the important ones are due to predation by introduced predators like cats and foxes, habitat destruction, changed fire regimes, and bounty hunting early on of a number of different native mammals. Many of Australia's surviving mammals have suffered really severe declines, and some of these species survive now only on predator-free offshore. Make it hard. I'm too tall. Predator-free offshore islands following complete extinction on mainland Australia. So in Australia, islands have acted as these really important arcs for threatened species, with 43 islands protecting 29 threatened mammal species from extinction. And this is because islands provide a refuge for many of the threats on the mainland, particularly predation by feral cats. As a result of this, we have these sort of repeated natural replicates that we can use to understand the genomic impact of island isolation across many different species in a comparative way. And importantly, islands are also really key sources for reintroduction back to the mainland of Australia, either as single source translocations or as mixed source translocations as part of genetic rescue. And here's just one example to represent many cases like it. This is the booty, or the burrowing bettong. It's a threatened Australian marsupial that was once distributed across a really wide range on mainland Australia, which you can see in the grey on the map. It's now completely extinct from that distribution and only occurs on 3 very small offshore islands off the coast of Western Australia. And so, as important as islands have been for the persistence of many native mammals like the booty, we don't yet have a good understanding of what the genomic impact has been for these species that once had these very widespread distributions that have now collapsed down to these tiny remnant populations. Theory predicts that small populations will lose genetic diversity, but also potentially accumulate harmful mutations. And that can lead to mutational meltdown and extinction. But it's unclear whether this part, the accumulation of harmful mutations, is true, or at least it's unclear under what circumstances it's true. So if a population is large enough for selection to act, then harmful mutations might be purged by selection, but does selection act in really, really small populations like on islands, or is genetic drift more powerful? So I'm going to talk about a couple of questions today using examples from some of our work on threatened marsupials. So first of all, I'm going to give a little bit of an insight into the question about whether island populations have more deleterious mutations or genetic load compared to their mainland relatives, and in many cases they're now extinct mainland relatives, or whether genetic load is purged on islands, whether genetic load is predicted by population size, and what the implications are for genetic load during mixing or genetic rescue of island populations. So I'm going to talk about data from 6 different threatened marsupial species, pictured here. They're very cute. These are the ones that we have the best sampling for, so they can kind of act as example flagship species to answer this sort of general question. And these 6 species occur across a variety of different historical mainland ranges. So across the top 3, those species have quite a widespread historical distribution that they're largely now extinct from or completely extinct from. And along the bottom, those 3 species have more of a fragmented historical range. But in all cases, they, importantly for our questions, also occur on offshore islands, and importantly for their persistence as well. So we have a total of 15 natural island populations included in the data that I'm going to talk about. So first I'm going to show you some results from neutral diversity. So with 202 samples from 21 natural populations, including island and mainland populations, we see a clear pattern where we've got higher heterozygosity or genetic diversity in mainland populations. Those are the box plots shown in gold. And that includes extinct mainland populations that have now changed to red, hopefully. and that's compared to their relatives living on islands in blue. So this shows that we've got a pervasive loss of genetic diversity on islands, and in all cases that we've got here, we've got relatively high diversity in now extinct mainland populations. Overall, we see that there's a positive relationship between range size and neutral diversity, which is what we would expect. And I also think this is a nice example of how we can use museum specimens to benchmark genetic diversity in now extinct populations. And these extinct populations are going to be included in the downstream analyses for the rest of the talk. Now I'm going to talk a bit about genetic load or harmful mutations. And I'm just going to give a little brief explanation of the terms that I'm about to use, because I know different people use different terms that might mean the same thing in this space. So if you imagine a potentially harmful mutation like this missense mutation arises in a population, that might be hidden from selection in the heterozygous state. And I'm going to call potentially harmful mutations like that the segregating load. And we expect that segregating load is going to be more prevalent in a large population. But if that mutation becomes homozygous, it's now exposed to selection, and as populations get smaller, genetic drift might lead these sorts of deleterious mutations to go to fixation in a population. And I'm going to call that the fixed load. So when we quantify these two different factors in our populations using these genomic proxies for genetic load, what we see is that we've got an excess in segregating load in the yellow bars in all of the mainland populations, including extinct populations. And then in contrast to that, we've got an excess, or at least a slight excess, in all of the island populations in terms of the fixed load in red. But you can see that there's a bit of sort of variation across species and populations, so we wanted to know whether that was related to population size or other factors that we might be able to model. So using a mixed model approach, we found that segregating load was predicted significantly by population size, in that larger populations are more likely to have a higher burden of segregating deleterious mutations, but not related to divergence time or generation time. So this was an expectation. But in contrast, we found that fixed load was not predicted by population size or any of the other factors that we modelled. And so we think that this suggests that there's evidence for purging in island populations, so we're seeing no evidence of a higher accumulated fixed load in smaller island populations. So if I've got enough time still, I'm going to talk a little bit now about what this means for genetic mixing. So given that these island mammal populations are now really important sources for reintroduction back to mainland Australia, we want to know whether the sort of nature of them being pre-purged of deleterious mutations, what consequences that has for genetic rescue. So again, using the booty as a case study, we're going to look at some empirical data from genetically mixed populations. Australia is a really great case study for this because we have a long history of this learning by doing approach to genetic management. So we have a number of different case studies that we can draw on to try to understand the genetic consequences of mixing. And in the booty, we've got multiple different translocations that are mixed source from two different islands. So those are shown in the map in red. So we know that successful mixing of populations should increase genetic diversity, and we can confirm this in the booty by seeing that the mixed populations have elevated heterozygosity and they fall intermediately in PCA space. But what about genetic loads? So when we look at data where we've mixed the most divergent island populations together, so in this case Barrow Island mixed with Doré Island, we can look at the parental distribution of segregating and fixed load and compare that to the F1 and F3 generations post-mixing. And what we see is that mixing divergent island populations reduces fixed load and increases segregating load in the short term. And when we mix more closely related island populations together, we see the same pattern but slightly less pronounced. So in the near term, mixing of these pre-purged island populations will reduce fixed load and increase segregating load, so potentially selection gets another chance at genetic purging, assuming that we are fostering the right conditions for this. So in mainland sanctuaries, like where these booties have been reintroduced, that will involve proper predator exclusion and population growth. So I think I've probably just about run out of time now. So in summary, from this project, we found that there's high segregating load in extinct populations on the mainland, that long-term small populations on islands have evidence of genetic purging, and that genetic mixing of pre-purged populations decreases fixed load and increases segregating load, which will potentially allow for additional purging under population growth. Thank you to all these wonderful people and to the organisations that funded this work, and thank you to ESEB for the opportunity to present to you today. If I have time for questions, I can take them, but otherwise I can talk to you afterwards.
Well, thank you so much for this honor to be here, and I'm looking forward to sharing with you some of my PhD work studying an inversion supergene in deer mice. So adaptive evolution often involves changes in multiple traits, whether that's shell size and shape in snails, seed size and branching patterns in sunflowers, or color and striping patterns of the stick insect. And this really raises the question of how do new adaptive forms arise and persist over time? So to address this question in the Hoekstra lab, we focused on this really classic example of local adaptation that involves distinct ecotypes of deer mice. So the deer mouse is shown here. It's a rodent species found across North America with its habitat range shown here on this map. It's got really large population sizes and is locally adapted to essentially all of the major terrestrial habitats across the continent. And it's been long noted that there are some striking phenotypic differences found between locally adapted populations. And sort of some of these most pronounced differences are found between forest and prairie ecotypes of the deer mouse. And these phenotypic differences found between forest and prairie mice persist over time despite a well-known gene flow connecting the populations. In particular, the forest mice have substantially longer tails than the prairie mice, and these long tails are thought to help with climbing behavior in the forested environment. The forest mice also have substantially darker coat colors than the prairie mice, and these dark coats help the forest mice blend in with their soil substrate to avoid predators. What we find is that when we bring these mice from the wild back into the lab, these trait differences persist in the lab colonies, suggesting a strong genetic component to both the tail length and the coat color differences. So this raises the question of how do these trait differences persist over time despite these populations being connected by gene flow? So to address this, we first performed a forward genetic cross using the forest and prairie mice in the lab. So we crossed the forest and prairie mice to create F1 hybrids, intercrossed these F1 hybrids to create F2s, essentially shuffling these genomes and allowing us to map these traits. We performed quantitative trait locus mapping of both tail length and coat color. So here I'm showing the tail length QTL map. So along the x-axis here, we have position along the genome. And on the y-axis, we have the LOD score, or the strength of association between our genotype and phenotype. And the black line you can see is the genome-wide significance threshold. So for tail length, we found that there were 5 regions of the genome significantly associated with variation in tail length. And the region that had the largest effect on tail length was on chromosome 15, and this QTL explained about 13% of the total variance in tail length across our F2s. So we then did the same process using coat color, and for coat color, we were rather surprised to find only one strongly significant QTL that happened to localize to the same region of chromosome 15 as our largest effect tail length locus. And for coat color, this chromosome 15 QTL explains about 45% of the variance. So because both of these traits were mapping to the same region of chromosome 15, we wanted to explore this QTL further. So we zoomed in on the QTL map. Here I'm just showing the QTL plot for chromosome 15 with coat color in red and tail length in blue. And what we noticed is that rather than a QTL peak, this looked more like a QTL plateau, which could indicate something like a chromosomal rearrangement underlying this region of the chromosome. Indeed, when we looked at recombination events across our F2 hybrids, we found that recombination was nearly completely suppressed across the same region of the chromosome. And so we thought, okay, perhaps there's a chromosomal rearrangement occurring in this region of chromosome 15. Indeed, when we performed PacBio long-read sequencing of one prairie mouse and one forest mouse, we found this really large chromosomal inversion spanning the first 41 megabases of the chromosome. So this suggested that an inversion is strongly linked to these two adaptive traits, the tail length and the coat color traits. We next returned to our wild-caught populations to look at the frequency of the inversion in the wild. So here I'm showing our focal forest and prairie populations. These are found in the Pacific Northwest in Oregon State. And we also sampled mice across an environmental gradient across the Cascade Mountains, which change quite sharply from forested to prairie habitat. And then we genotyped these samples for the inversion. So here we're looking at the frequency of the inversion across this environmental transect. And what we found is that the inversion is nearly fixed in our forest population. It's at 90% frequency in the forest. It's completely absent in our prairie population, and what you can see is that it changes in frequency as you go across this environmental gradient. So this suggests that the inversion is associated with forested habitats, consistent with it playing a role in local adaptation. So just to zoom out for a second, why is an inversion important in the context of this local adaptation? Well, when an inversion is heterozygous, it will suppress recombination, and that's because in order for recombination to occur within these inversion heterozygotes, inversion loops like this one will typically form, and when you get recombination within an inversion loop, these recombinant gametes tend to be inviable due to large duplications and deletions. So in our case, where we have this inversion linked to both the tail length and the coat color traits, because recombination is suppressed in the heterozygotes, the inversion is keeping the tail length and coat color mutations together. So as these populations are connected by gene flow, the inversion is ensuring the co-inheritance of the long tails and the dark coats in the forested population, despite constant migration from the prairie. So this means that the inversion is acting as a supergene, where it's really helping to facilitate the maintenance of these distinct forest and prairie ecotypes. So finding this supergene allowed us to then address a sort of long-standing question, which is how do inversion supergenes arise? And we're thinking about this really with two models. First is the capture model, where you can have adaptive loci segregating in your population or your species. And then an inversion arises and happens to capture some sort of lucky combination of adaptive loci. And in this model, we're also including the possibility that the inversion breakpoint itself causes the adaptive phenotype. In contrast, what we're calling the accumulation model is one in which the inversion occurs in your population, and then over time, adaptive mutations accumulate on this inversion until it becomes a supergene. So we wanted to distinguish between these models. And to do so, first we set out to actually identify the coat color and tail length loci within the inversion. So to do this, we performed a controlled study of the inversion's phenotypic effects, where we isolated both the inversion and non-inverted, or the standard haplotypes, on a consistent forest genomic background. And then we crossed heterozygotes to create this congenic mouse line where we have sibling offspring that just differ at the inversion locus, allowing us to control for both genetic and environmental effects. So we use this congenic line first to look at coat color more carefully. And for coat color, what we found is that when we compare our homozygous congenic mice and we look at pigmentation in the mouse hairs, we find that the inversion almost completely removes the light pigment band that's typical in mouse hairs. We then used these congenic mice to perform RNA sequencing experiments. So we looked for genes that were differentially expressed in the skin during hair growth of both the inversion and standard homozygous genotypes. And we found within the inversion there are 150 coding genes. Of these, only 9 are differentially expressed in the skin. And of those 9 genes, 2 of the genes are known to be expressed in the hair follicle using data from lab mice. So this led us to some candidate genes, including our top candidate gene at the moment, NKD2, which is a gene that acts upstream of the mouse pigmentation pathway. So we're now following up on this gene with functional testing. We also used the congenic mice to look at tail length more closely. And for tail length, we could use X-ray data to look for differences between the inversion and standard haplotypes. And what we find is that the inversion increases tail length by increasing the length of the tail vertebrae, but not increasing the total number of tail vertebrae. And similarly, we could use differential expression analyses to find genes within the inversion that are differentially expressed during tail vertebral growth, of which we found 12 within the inversion. We also took advantage of populations in which the inversion does not have effects on tail length to actually find map the causal tail length loci within the inversion. And what we found is that the inversion maps to this, the tail length loci maps to this relatively narrow region within the middle of the inversion, which suggests to us that the tail phenotype is not caused by the inversion breakpoint itself, but rather by mutations that are found in the middle of the inversion. Further, we found that there are 20 genes within this relatively narrow region within the inversion, only one of which is differentially expressed in our developing tail vertebrae. And this gene is NPR3, a gene that's known to cause increased vertebral length in lab mice. So we're excited about this gene and following up here with some functional testing. So we next looked at the inversion's phenotypic effects across populations, looking at the whole species. So here I'm showing a map of the inversion frequency across the species range, with the inversion in green and the standard haplotype in tan. What we found is that the inversion is primarily localizing to the western portion of the species range, and there are a number of populations in which the inversion is segregating. So we focused on 4 of those populations and phenotyped those populations for both coat color and tail length. And what we found is that the inversion consistently shows effects on coat color across all 4 of these sampled populations. But interestingly, for tail length, we found this population D, we're calling from New Mexico, in which the inversion showed no significant effects on tail length. So this suggested to us that the inversion is acting as a supergene in some, but not all of the populations in which it occurs. We could then use these populations to start to reconstruct the evolutionary history of this inversion supergene. So here I'm showing a phylogeny of using SNPs found within the inversion region, and we have the standard haplotype for these 4 populations on top and the inversion haplotype on the bottom. What this phylogeny suggests is that the inversion likely arose some time ago and either quickly accumulated, or captured the coat color mutation. But because there's this population D, which doesn't show an effect of the inversion on tail length, we suspect that the tail length mutation or mutations arose sometime later, such that only some, but not all of the population, such that the inversion is acting as a supergene in only some, but not all of the populations. In other words, we think that the inversion likely became a supergene over time through the accumulation of these tail length mutation or mutations. So this is supporting the accumulation model of inversion supergene evolution. Okay, so in conclusion, we were really interested in understanding how these distinct forest and prairie ecotypes of deer mice are maintained over time despite ongoing gene flow. What we found is that an inversion is playing this very important role in facilitating local adaptation through linking these multiple adaptive traits, the tail length and the coat color traits, together and acting as a supergene. We further found that inversions need not capture a particularly lucky combination of alleles, but can actually become a supergene over time through the accumulation of adaptive mutation. So I'll just end by saying I focused today on this one inversion supergene, but we've actually found that these really large megabase scale inversions are quite common across the deer mouse genome, with examples shown across the genome here. And we have evidence that multiple of these inversions are contributing to local adaptation, providing a really nice system for studying inversions. And so I'm now continuing much of this work as a Lewis Sigler scholar at Princeton. So with that, I'd like to thank my collaborators on this project, in particular Emily Hager, Shwanan He, Brock Wooldridge, and my advisor, Hopi Hoekstra. And if there's time, I'll take questions. Yeah. Okay.
Okay. I don't know where to start because I can't see my slide from here. All right. So yes. So I'm from Sierra Leone, and I try to represent the map of Africa with Sierra Leone in it on the point there. And that's because this is the most questions I receive around Europe and in Western countries. Like, is Sierra Leone in the Caribbean? So no, some geography lesson. Sierra Leone is in the west coast of Africa. And that's where I come from. And okay, great, it's going. So yeah, so I was born and grew up in a very small village in the northern part of Sierra Leone. And it's a small village where no basic amenities existed at the time. And so all you think of pipe-borne water, road leading to these communities were not there, so vehicles could not even enter these villages. No electricity and nothing you can think of, no school. So my school was like 5 kilometers away from my village. And at the age of 7, I started going to the school, and I had to walk like 5 kilometers every day to and fro, and I had to go barefooted and without food. And so that was what I had to go through for about 6 years of my first 6 years of primary school, because then going to school means that you get a shoes. The first shoes will be given to you at the point where you enter your junior high. So it was a kind of trophy to go to school and get a shoes at junior high. So it was the determination to get there. So, and then, but like in the kind of situation you live, and everything kind of seemed like horrible for you, but that was like what we had and nothing to compare with, and so we were happy as kids growing up. But then the war broke out in 1991, and then rebels were attacking the country left and right, here and there. We had to always run from one village to another for refuge, and so schools were disrupted for years, and so we had to leave the village and go to the capital city where it was a bit safer and to continue school. But while arriving at the city, at just 2 months later, the rebels attacked the city. And unfortunately for me, I was abducted by the rebels. And while they were retreating back to the jungles, I was taken as captive and also be trained as a child soldier to reinforce their depleted squad during the fight in Freetown. Thousands of young kids were taken, and so I was trained, and I had to go through this over a year in the jungles to fight and survive against all the artilleries that were coming from conventional warfare from the West African forces that were fighting against defeating the rebels in the jungles. So, yeah, but I had to go through that, and to cut long story short, I survived it, and I was able to escape, and I went back to my family, who had already organized my funeral service because they thought I was dead. And then getting there, they was happy to receive me, and I had to continue school. My passion was to go to school, and even though there was nothing, I had to do all the worst things to get money to go to school. I had to beg on the street. I had to do all that kind of stuff to go to school, until I was lucky one day to meet one U.S. missionary who had compassion in me and saw my motivation, and then paid the remaining years of my high school, and then up to university. So at university, she paid up to I finished university, and the rest is history. I have gone on to study in Nigeria for a master's degree, and then in France for a master's degree, and now in Finland. Very cold. Yes. Needless to say, you'll know. So in Finland, the research group where I do my PhD work, in Finland uses Atlantic salmon as a model species for studying the genetic basis of life history variation. But they have been particularly interested in studying factors affecting variation in salmon maturity age, the variation that are there in the difference in maturity age. And so this was due to the positive association with reproductive success that's trade off with survival probability. So some background is that after a salmon has hatched in the rivers, it spends some time feeding and growing until it eventually migrates into the sea, where it continues to feed and grow, and then until it decides to go back, to swim back to its home river to reproduce. And so the time it spent at sea before returning is what's called the age of maturity. And there's a huge difference in this, and there are those that return after 1 year, there are those that return after 2 years, and 4, as the case may be. But there's also a huge variation in the size at which they return. So, so in trying to understand this background and this variation that is being seen in the size and the age at which they return, a genome-wide association study was done. And what they found was that one genomic region explained a huge variation in the maturation time. And that's genomic region is the the candidate gene was the VGLL3. And the VGLL3 explains some about 39% of this variation. And so this VGLL3 has 2 alleles, and the L for the late maturation and the E for the early maturation. And then, so looking at growth and maturation probability in a lab-reared Atlantic salmon, one of our lab study found that individuals with the E allele tended to have higher body condition, and meaning that they were heavier for their length, or otherwise they were fatter compared to the L alleles. So what this result suggested was that it might be the differences in body condition that drives the differences in maturation. And so we thought maybe that VGLL3 might be affecting early resource allocation to influence maturation. And so going from finding some body condition and gene expression that has been done by other colleagues in the lab, I wanted to explore how VGLL3 affects behavior. And so I designed a simple study to measure aggressive behavior, exploratory behavior, and risk-taking behavior on salmon with different VGLL3 genotypes. And so the aim of my study then was to see if this condition that exists in later in life already exists at the juvenile stage with regards to this VGLL3 locus. My hypothesis was based on the fact that the pace of life syndrome explains that coordinated suite of life history traits and behavioral characteristics evolve together. Specifically, they state that the pace of life, the fast pace of life, in the fast pace of life, organisms tend to exhibit behavior. They actually mature quickly, and then they reproduce at an early age, and they also have a shorter lifespan. So these organisms normally tend to exhibit behaviors which are advantageous for rapid resource acquisition and early reproduction, such behaviors as aggressiveness, exploratory, and boldness. But in the slow pace of life, organisms mature slowly, they reproduce later, and have longer lifespans. So these organisms normally tend to be very cautious, they are less aggressive, and they're risk adverse, focusing on long-term survival and sustained reproductive success. So for my hypothesis, it's kind of like that, with the framework that has been presented by the pace of life, and with prior studies where early maturation genotype tended to have higher body condition, so it's kind of like that the individuals with the early maturation should exhibit a fast pace of life. So that was what I tested. And in my study, there were 2 main trials I tested. So one was the aggressive behavior. And in the aggressive behavior, I used 7 families of salmon fry, where all offspring had the early maturation genotype, that's the VGLF3EE, and 7 families with the late maturation genotype. These were reared in semi-structural system in tank where tanks receive similar treatment across tanks. So for our behavioral trial, we size matched 4 individuals, which were placed in a tank, and then we were able to score their behavioral differences. So this was done for the aggression behavior, and for the activity behavior, we placed individuals in tanks, which were randomly selected from their holding tanks and then placed, and then we observed their exploratory behavior in an open environment and also in the presence of a novel object. So I'm not sure I can play this without the remote here. Okay, so I can't. But what I wanted to show you there quickly was the fact that individuals were behaving in different ways, in such that one set of behavior was that once the novel object was introduced, they attack it immediately. The other was like just wanted to go away and just stay far from it. So for our results in the aggression trial was that we saw that the individuals with late maturation were more aggressive, and that for the exploratory behavior, we found one specific, that there was a sex difference in their behavior, in such that the males of the late maturation were the quickest to leave, to move. But this results actually, okay, then we also found that individuals in the novel object trials were moving very fast. They were moving very fast, especially for those with the late maturation. And all these results were in line with the previous study of the aggression study, where individuals with the late maturation were more aggressive. So in conclusion, we see that there's a proactive behavior like the aggression and risk-taking exploratory behavior. They are very energy-consuming, such that if these are persistent over time up to their adult age, they would not mature earlier, because energy that would have been spent for reproduction are now spent into being very active. So, and then that there was also the sex-specific differences that we saw, in such that these behavioral differences are shaped by distinct evolutionary pressure and reproductive strategy that each sex face. For example, male might have exhibit higher boldness and exploratory behavior due to their need to compete for mating, but also for resources and to defend their territory, while female might be more cautious to optimize energy use for reproduction and avoid predation. Thank you for your attention, and thank you, ESEB, for this opportunity.